So we now have completed Chapter 12, and Chapter 12 actually is split into three presentations, 12-1, 12-2, 12-3, that are available on the streaming video on the WebCT site. And, of course, the lectures themselves are posted, as you know. After completing thereby our consideration of the peptides, that included a lot of discussion of neurotransmitters, of course. Now we move to Chapter 5, which is covering neurotransmitters separately, so it's dedicated to that. So let's switch to the computer and work with the Chapter 5 on neurotransmitters. I'm just going to set my pointer here. Always forget to do that ahead of time. And I realize you can't read the text here, but this is posted as black lettering on a white background, and you can either have it open at the time or printed in front of you. And much of this chapter is from the textbook, unlike some of the previous lectures so far. And what we're considering are the components of Table 5.1. And we've mentioned these so many times, I'll just quickly go through them. It's got to be in the presynaptic neuron. We've got to have evidence for precursors and syntheses, biosynthetic mechanisms in that neuron. You've got to be able to activate the neuron either by increasing the transsynaptic excitation by stimulating afferents going to the neuron, or directly exciting the dendrite or the cell body, and see that there is release of physiologically significant amounts at the synapse, at the end terminal. And the afferent stimulation to excite the neuron, or if you can naturally recruit that excitation of the circuit, should produce responses very similar to when you stimulate the neuron or the population of neurons themselves. You've got to be able to add it at the synapse, either by injecting it into that part of the brain, or maybe you're just looking at the electrophysiology, as we saw in the discussion in Chapter 12, and see that it has identical effects as the stimulus response, that depending on what response you're measuring, whether it's the behavior of an animal, the release of hormones, or electrophysiology. Got to have the receptors, so we know we're building up to a discussion of the receptors. They've got to be close to the site of release, obviously. And for neurotransmitters, we know that we are talking about transsynaptic effects, so it is released from the presynaptic terminal, and receptors present immediately on the other side of the synapse on the postsynaptic membrane. And the interaction of that substance under consideration and the receptors has got to produce some change in the postsynaptic cell. Here we're talking about neurocrine type of chemical messenger processes, neurotransmitter functions, in other words. So we're talking about the membrane permeability, just as we have discussed in Chapter 12, and we've got a better feel for membrane permeability and the effects on the membrane potential, whether it's altering resting membrane potential or actually generating an action potential. We have a better understanding of that now, and we'll continue to improve our understanding as we proceed through our discussions in the semester. So there's really no message carried in this sense by a neurotransmitter unless it changes the permeability to an ion and alters the membrane potential in the target neuron. I'm not saying it has to cause an action potential, just has to alter it. Specific inactivation mechanisms are expected, and it's got to be within a reasonable time. So if you were testing it and it took an hour to eliminate a neurotransmitter, that you had added a certain amount of the transmitter, I guess the half-life then would be 30 minutes. That's still pretty long for these smaller molecules. For peptides, it's still pretty long as well. But it's got to be physiologically relevant time frame that you get the inactivation. Agonist and antagonist effects are obvious, and you'll find in this chapter, toward the end, there are tables listing many different receptor subtypes and many different agonists and antagonists for the different transmitters and the different receptor subtypes. That won't be required knowledge or anything that you would be tested on, other than when we begin to look at some specific research journal articles where they may have used a particular drug. That's when we'll identify it and its mechanism of action. Then it's added to our list of must know, or you're expected to know and recognize. So in other words, we want to start moving away from any feeling that we've got to try to memorize this long list of terms and other columns of information about them to having a particular graph that we viewed and can associate the drug with in our mind and begin to say, okay, this drug did that, so it must be an alpha-2 antagonist. So some means of linking it together. So it's more of a working framework of information beyond just trying to memorize. So there are many different categories of transmitters, and I realize I may not be providing any new information for anyone, but what we're trying to do is provide the same information so everyone knows the framework of basic information that we want to have in mind to proceed to more detailed information as we proceed through the course. So if it's a quick refresher for you, great. If it's the first time, great as well. You got a little work to catch up to other people, but it shouldn't be a real problem. So we'll talk about amino acid transmitters, excitatory and inhibitory, the cholinergic neurotransmission, one of the earliest that was discovered, as well as the monoaminergic, and then a little bit about peptide transmitters, and then very little bit about putative neurotransmitters, just to let us realize that there are new categories being discovered ever so slowly, but we are reminded that we don't know everything at present. So we start first then with the amino acid transmitters. So you'll see that this is information that I have extracted from an introductory human anatomy and physiology textbook, and it augments and supplements what is in our textbook, but it still should be basic knowledge that we have from taking a series of biology courses to get our degree in biology, which I'm expecting everybody probably has to be in this graduate level course. So the first of the type of amino acid transmitters that we consider is the gamma aminobutyric acid, or GABA. So I like this because it shows you the structure of these. You should be able to recognize these, so maybe drawing them every now and then as it comes time to take the test would be a good idea so that you can remember. If you recall, with a carboxylic acid, you have an alpha, beta, and gamma carbon molecule. So gamma aminobutyric acid, butyric is for carbon, and carboxylic acid is called an acid. So if you remember your organic chemistry, a little bit of biochemistry, it helps too. So this is our chance where we bring in just a little bit of organic chemistry and a little bit of biochemistry to talk about these molecules. So don't get too concerned about it, but also don't disregard it. It can help you if you just push back and go, oh yeah, 4 carbons is the butyryl, and the COOH is a carboxylic acid. If we start there, we have an alpha, beta, and gamma carbon moving away from it. So this is the gamma carbon with the amino group on it, so easily gamma aminobutyric acid. This is the major inhibitory neurotransmitter, fast-acting neurotransmitter in the nervous system. Its primary mechanism of action is through the GABA-A postsynaptic receptor. That is a ligand-gated ion channel specific for chloride. So when GABA binds to its GABA-A postsynaptic receptor, opens up chloride influx, more negativity inside the membrane, it becomes hyperpolarized. So this is found throughout the CNS, especially the hypothalamus. Major break, we've already talked about differences in activating neurons, or even if we extend it to the release of hormones, we can either play with our gas pedal or our brake. We can have the gas pedal on and increase the brake, or we can, and that would slow things down, or we can just let up on the gas pedal. So the gas pedal is the excitatory drive, the brake is the inhibitory drive. So we always have some excitatory tone and some inhibitory tone, whether we're thinking about release. Think of the somatotrope with GRH or GHRH as the gas pedal, and somatostatin as the brake on it. So the balance between those two is determining the level of secretion and synthesis of growth hormone at any point in time. So you've got some tone, some baseline level homeostatic tone of gas pedal and brake in that example. And if you want to increase synthesis, you can either decrease synthesis and secretion of growth hormone. You can either decrease the amount of somatostatin, so you're letting your foot off the brake pedal, and the amount of gas pedal you've got going is now accentuated. Or you can just increase the amount of growth hormone releasing hormone, step your foot on the gas pedal, and override the inhibition and increase the release and synthesis. So we need to be thinking that way when we're talking now about excitatory and inhibitory neurotransmitters. So it's really throughout. And as I said, it's the principal inhibitory one. There's also presynaptic inhibition mediated through this at the axoaxonal synapse. So we've talked about an axoaxonal synapse. If you can hyperpolarize the target axon membrane, the postsynaptic membrane, an action potential coming down is going to be, can even be eliminated if you hyperpolarize that axonal membrane enough. So it's a very powerful way of inhibiting things, inhibiting release, at an axon terminal. We know that alcohol can have some effects, the drugs such as benzodiazepines like Valium, and if there's anything that blocks the synthesis or release of GABA, it can induce convulsions. So something else that isn't listed here are the barbiturates. So barbiturates are actually used to activate GABA receptors or enhance the activation of GABA receptors to prevent convulsions or to stop convulsions. Next, then, we consider the glutamate as an amino acid transmitter, and it is generally excitatory. It has the direct action. It's got a variety of receptors. We'll talk about its receptor subtypes, at least for the NMDA type. The N-methyl D-aspartate is very closely related to glutamate in structure and can bind to the glutamate receptors and helps us to characterize them as NMDA or non-NMDA, and we'll see that as we proceed. So here's the structure of glutamate. It is an amino acid. So here's the amino end, and here's the carboxylic acid end of the amino acid. And it has a carboxylic acid group on the gamma carbon. And this should immediately make you go, wait, that looks very much like a molecule we just looked at. Carboxyl group, alpha, beta, gamma, and with the NH2 here, decarboxylate, glutamate, and what have you got? GABA. So the enzyme for the synthesis of GABA is glutamic acid decarboxylate. Decarboxylate glutamate, the primary fast excitatory neurotransmitter in the nervous system. You decarboxylate it, and what have you got? Gamma aminobutyric acid, the major fast inhibitory neurotransmitter in the entire nervous system. So it's kind of interesting. So obviously, if you increase glutamate levels or activation of glutamate receptors, you're going to induce convulsions. So if you take your foot off the gas pedal by blocking GABA's effect, you can induce convulsions. If you step on the gas pedal, take your foot off the brake, I hope I said, for GABA, by blocking its effects, you can induce convulsions. If you step on the gas pedal by increasing glutamate activity, you can overexcite neurons as well and induce convulsions. But it also can push to a level of excitotoxicity, overexciting neurons and causing the death. So neurons are literally stimulated to death, such as with ischemia, the loss of blood flow, and the anoxia or hypoxia used to the associated with that because of the dramatic decrease, the hypoxia or loss of oxygen. So it's a very important neurotransmitter, not just for neural function, but clinically as well. Then the glycine is the other amino acid transmitter that we're going to talk about. So what you're seeing now is that these two examples are amino acids used as neurotransmitters. We can further subcategorize these as modified amino acids like GABA and histamine and the catecholamines. So there are some differences. Gamma aminobutyric acid is a slightly modified amino acid, but glutamate and glycine are not altered at all. They just are packaged into vesicles so that their release at a synapse is coupled to the firing rate of the neuron of origin. Therefore, you've got the exocytotic release coupled to the firing rate of the neuron, so it is a true signal. Again, to have a true signal, we've got to have a way of sequestering a readily releasable pool, get it out into the interstitial fluid and to the target cell, so the target cell feels something new or realizes there's something new through receptors, and then degrade it and remove that signal so that it's episodic. So something new, either higher or lower than where I was before, and my receptors are transmitting that signal to me. Glycine, on the other hand, is generally inhibitory, and it's found predominantly in the spinal cord in most cases. This is the receptor that strychnine targets, and it blocks the glycine receptor, therefore taking the foot off the brake pedal, disinhibiting the target cell. So it can result in uncontrolled convulsions and respiratory arrest, so really quite important, and obviously within the CNS. Then we come to other neurotransmitters, the cholinergic neurotransmitter, acetylcholine, of course. Acetylcholine, its structure is shown below here, so we'll look at its synthesis in just a little while. I guess this is the acetyl group down here, or am I wrong? Anyway, I guess this maybe is the choline and the acetyl group. My organic chemistry is not so good, is it? So correct me if I'm wrong, and if you know better than me, send me an email, and so I will train me again, so I'll know myself. When we look at the synthesis, we can straighten that out. So this is excitatory to the skeletal muscle, also to the adrenal medulla, and its direct action is through the nicotinic receptors, and there's an indirect action via second messengers through a muscarinic receptor. So I will expect you to know the distinction between the nicotinic and the muscarinic receptors. In that, at least we will talk about the nicotinic receptors at the adrenal medulla and how that can be modulated by other molecules, by other peptides. So there will be an article when we talk about the receptor mechanisms related to the activity of the nicotinic cholinergic receptor at the adrenal medulla. These are ligand-gated sodium ion channels, so when nicotine, as the agonist, binds to the receptor, or when acetylcholine binds, opens the channel, sodium influx occurs, more positive ions on the inside of the membrane. We depolarize the membrane, so it's an excitatory event. Very important in the CNS, in the peripheral nervous system, as I mentioned, even for the autonomic nervous system. And I'm not going to go, whoa, I pushed the wrong button. I went all the way to the end. Sorry. Where'd you go? Oh, wow. Maybe I should push home and go down. Sorry about that. Holy moly. So you can read the comments on this. It's very important in cognitive function, but we are thinking of it for operating as a neurotransmitter in the hypothalamus, and we'll see it as important there, and as well as a transmitter at the interface between the sympathetic nervous system and the adrenal medulla, and of course in the entire autonomic nervous system. It is used by the preganglionic neurons that are relaying central nervous system or CNS information out to the postganglionic neurons in the periphery. And we'll build on that storyline as we proceed. The biogenic amines are next, and these include the catecholamines. The catecholamines include norepinephrine, dopamine, and epinephrine. We'll look at the biosynthesis of these. So I use first the norepinephrine and dopamine here just to show you the slight differences between these. In this case, we start at the amino group to identify the carbons. This is the alpha carbon. This is the beta carbon. So if we have a molecule, an enzyme that will hydroxylate the beta carbon of dopamine, we would call it dopamine beta hydroxylase. So the enzyme that converts dopamine to norepinephrine is dopamine beta DBH, dopamine beta hydroxylase. So DBH is the acronym for it, and we'll see that later. So you should be able to recognize the structure of dopamine, and that the beta carbon with the OH on it lets us know that that is norepinephrine. So I'm not going to read through these columns on all of the information. We'll cover more and more of this information as we proceed. Norepinephrine is very important in the hypothalamus for neuroendocrine transduction events, as is dopamine, and we've already seen dopamine acting as a prolactin inhibitory factor. So the tuberoinfundibular dopamine neurons actually release dopamine into the hypothalamo-pituitary portal vessel system for targeting receptors, targeting dopamine receptors in the anterior pituitary. So the catecholamines are one type of biogenic amine. Another type of biogenic amine are the indolamines. Your textbook has these terms in the spelling, so therefore I'm not providing those. The indolamines include serotonin and melatonin. Serotonin is shown on the slide here. And then histamine has its own separate category as a biogenic amine. So there's catecholamines, norepinephrine, Let me list them in the synthesis pathway. Dopamine, norepinephrine, and epinephrine. Dopamine can be converted to norepinephrine. Norepinephrine can be converted to epinephrine. And serotonin and melatonin, and those are interchangeable. In the pituitary gland, serotonin gets converted to melatonin, and melatonin can be converted to serotonin. The serotonin, we see the molecule here, so it's very different. It has this 5-member ring within it that is very distinctive, other than the 6-member ring that we saw, the phenol ring basically, that we saw in the catecholamines. Since you recognize your amino acid structures immediately, like I do, you certainly view that this comes from a different amino acid precursor than does the catecholamine group. Tyrosine is the precursor to the catecholamines, so somewhere on here would have to have been the carboxy terminus. We see the amino terminus. The carboxy terminus would be on there, so we're going to have to decarboxylate. Remember the amine precursor uptake decarboxylase, common origin of many of these cells. So we have to uptake an amine, tyrosine, an amino acid, decarboxylate it, then convert it to dopamine, and then it can be converted onward. The amine precursor or amino acid precursor for serotonin is tryptophan, and the tryptophan has both the 6 and the 5-member ring within it, and I don't expect us all to recognize that and remember it right off, but it rings a bell when we hear it, right? We see that it's mainly inhibitory, and I don't even want to even think about that. We should just scratch that idea. It has the effect that it has in the location where it is studied carefully, and the best way to go about it is to say, I'm not really sure unless I know the specific studies for that part of the brain or for that neuroendocrine system. It gets pretty tricky. There are many, many serotonin receptors. There is one that can act through a direct action. The 5-HT3 receptor is a ligand-gated ion channel. But all the other serotonin receptors, and there's a dozen or more now, are metabotropic receptors, and therefore would be considered more so to be neuromodulatory in function than neurotransmitter. And serotonin does act extrasynaptically, so it is a prime example of neurotransmitter, neuromodulator, and should just be considered as a neuroregulator, I guess, because it is both a transmitter and a modulator. It's found throughout the entire central nervous system and peripheral nervous system, very important throughout the body, as we know. Many of the antidepressant drugs, the selective serotonin reuptake inhibitors, target the serotonergic system. Prozac is a prime example. So this just reminds us of the importance of this transmitter throughout the CNS. We, of course, will be focusing on its function in the hypothalamus and the impact on certain of the neuroendocrine axes. But it also reminds us of the linkage between psychogenic components of the organism, internal environmental changes, carries sensory information indirectly, as well as being associated with stimuli from the external environment thereby. So it helps to integrate our emotions, behavior, and neuroendocrine functions as well. Down in the spinal cord, it contributes to the autonomic nervous system, so also our somatic motor and our visceral motor, blood pressure and heart rate, components of reaction to a situation, change in the external environment. Did you have a question? Oh, okay. And then histamine is the next one. There's not a lot known about it. If we have allergies, we know that when our mast cells release it, it gives us many of our allergy symptoms, including association with inflammation, because it's a powerful vasodilator. But we also know that if we're taking antihistamines that aren't designed to be restricted at the blood-brain barrier so it doesn't cross into the brain, then we don't get drowsy. But if you take an antihistamine that does cross the blood-brain barrier, you can become very drowsy and a little bit disoriented, might even go to sleep. So quite obviously, histamine plays a very significant role within the brain and the CNS in alertness, attentiveness, and the sleep and wakeful function of the nervous system. And if you don't believe it, just take Benadryl and see what happens, right? Well, let's continue with the discussion of these. The monoamines then include the catecholamines, the indolamines, and the histamines. So they have only one amine group on them. So the catecholamines, of course, are the dopamine. Here it's NA for noradrenaline and A for adrenaline, but it really should be norepinephrine and epinephrine. The adrenaline and noradrenaline are no longer used, even though the receptors are still called adrenergic receptors. The terminology used is norepinephrine and epinephrine, and they are synthesized and released in the adrenergic pathways that we'll look at. The indolamines, serotonin and melatonin. There's a small number of serotonin cell bodies, neuron cell bodies, and a midline part called the raphe. Raphe actually means midline. In the reticular portion of the medulla, the pons, and the upper brainstem. So this is actually part of the reticular activating system, and contributes to sleep-wakeful function and reduces emotional behavior. The histamines, primarily in the hypothalamus and the limbic system. The neurons that are synthesizing histamine are located almost exclusively in the hypothalamus and the posterior hypothalamus predominantly. So when we find histamine in other areas, such as in the limbic system and the cortex, those are the axon terminals, the projections of those hypothalamic neurons. There are many peripheral actions of histamine that we're more familiar with, as I said, than the central nervous system. And there still is very little research performed on the central histaminergic system, and it's kind of shocking. There are a wide variety of peptide transmitters, substance P, neurotensin, somatostatin, and enkephalins. We've talked about many with our discussion of neuropeptides 1 and 2 already, and I just want to spend a bit of time showing a few examples. Here we see the endorphins, the dynorphins, and enkephalins. Enkephalin is the one that's illustrated here. So 1, 2, 3, 4, 5 amino acids. This is a pentapeptide. So these are rather small peptide transmitters. It's generally inhibitory and has actually more of a neuromodulatory function because it acts to a second messenger system. Widely distributed opioid receptors throughout the entire nervous system. The impact on pain is discussed in the last part of Chapter 12, but it does have significant effects in the hypothalamus on neuroendocrine function, as also discussed in Chapter 12. The tachykinins include substance P. We've talked about substance P. It's a prime example as a neurotransmitter, a peptide neurotransmitter from Chapter 12. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 And decapeptide, is that what it would be? So 11 amino acid residues, so it's a rather large molecule. We've talked about somatostatin. We see that it has a characteristic three-dimensional shape. We see a cysteine and a cysteine amino acid here. So we know that that's where we're going to have the disulfide bond linkage. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 amino acid residues for the somatostatin. And we'll use somatostatin at one point to talk about post-translational processing, but we won't deal with it now. There are many different forms of somatostatin. Generally inhibitory as a peptide transmitter or neuroregulator, if it's a modulator, because it acts via a second messenger system. Again, if they act transsynaptically, they can be considered as a transmitter. So that's the ultimate distinction between transmitter and modulator, as you recall. Here's cholecystokinin. We've heard this one before. 1, 2, 3, 4, 5, 6, 7, 8. So it's an octapeptide, and it has a sulfated amino acid residue, a sulfated tyrosine. So it's a bit distinctive. So somatostatin has a disulfide bond. Cholecystokinin has a sulfated residue. So if there's a post-translational modification of these, I encourage you to give it some attention. Some of the putative neurotransmitters, like the prostaglandins and the endogenous benzodiazepines, so far do not meet a satisfactory number of the criteria to be categorized as a neurotransmitter, so they're called putative neurotransmitters, and I won't mention anything more about those. Now let's spend just a bit of time reviewing the synapse and some of the functions there. We build on this as we cover different chapters, so the whole picture continues to come together. My synapses didn't seem to be working when I spelled communicates here, so forgive my misspellings there. We know the function of the synapse. It's the neurocrine, the structure required for neurocrine chemical messenger signaling. And there can be different types of these synapses. An axon terminating on a dendrite is the axodendritic synapse. An axon terminating on the somatic, the soma or the cell body is the axosomatic type. Axoaxonal synapse, one axon onto another axon. We see those illustrated in this slide. The cell bodies, the main part, there are different processes coming off the neuron. Can be dendrites for the receiving or the input side, or the main single output type of process, the axon. So the axodendritic synapses, an axon onto the dendrite, axosomatic onto the cell body, axoaxonic here on the initial segment of the axon. But the other type we had mentioned was axoaxonic, where it would be one terminal onto another terminal, which also does occur and becomes very important. So here's a scanning electron micrograph showing us the axon, and it actually does branch quite profusely as it approaches the target cell. And this is onto the cell body, so these are axosomatic synapses. And they can either be excitatory or inhibitory. So the excitatory is going to depolarize the cell membrane, bringing resting membrane potential closer to threshold, as we recall from the prior lectures. These are very commonly found on the dendrites. The inhibitory are, of course, going to hyperpolarize the membrane, and not always hyperpolarize necessarily. We should say that it's just going to increase the negativity inside the cell membrane, and will hyperpolarize if it is pushing the membrane potential below the resting membrane potential. This usually occurs at the cell body. And in considering neuroendocrine transduction in the hypothalamus, it's probably safe to say virtually all of the neuroendocrine cells have both glutamate and GABA input, excitation and inhibition. And then other neurotransmitters are going to modulate that gas pedal and brake to some degree, whether it's peptide or the classical transmitters that we just mentioned. So I'll end with this slide showing this illustration from the text, so the dendrites. And the one key distinction on this one, other than typical type of axodendritic, axosomatic types of terminals, the inhibitory synapse shown here with the ovoid type of synaptic vesicles and not a prominent postsynaptic membrane or density. The excitatory synapses having the circular or definitely not the oblong, elongated type of vesicles, and a distinctive postsynaptic density. It's actually within this postsynaptic density that many of the glutamate receptors are embedded and contributing along with other proteins to this density. So there's distinctions between excitatory and inhibitory at the structure level, but the one that I want to point out here is the one on the spine, these little extensions of the dendrites, so the axospinous input, and this is quite common on neuroendocrine cells as well. But typically what happens here, they're only showing an excitatory synapse on the head of this spine, as it's called. On the neck of the spine down here is where you usually have the brake pedal inhibitory input. So there's a large amount of room for integration at the neuron. And for neuroendocrine cells, it simply gets turned into firing rate, and firing for any of the neurons, it turns into firing rate, and the firing rate determines the quantal release or the level of release. Whenever we talk about release or secretion, we know that it has similar impacts on the expression at the level of the genome, in this case in the soma. So I appreciate your time and attention. And for those of you in the viewing audience of the future, this is the eve of Hurricane Rita, September 23rd, 2005, about to hit the Texas coast. So you can tell us how the future has changed because of that. I hope you all have a nice weekend. See you again soon. Bye-bye.
Be careful. Have a nice weekend.
You too. Bye-bye.